I'm Xiaojie, so I'm actually a postdoc in Jonathan Weissman's lab at MIT. So first of all, I would like to give a big thanks for the STOC organization inviting me to give this presentation here. So for today's talk, I'm gonna tell you my story on Dynamo for predictive modeling of single-cell genomics. In the last few minutes, I'm gonna also briefly talk about another story on Spatio, a new toolkit developed by me, BGA research and others for quantitative modeling of spatial transcriptomics. So for many years, I have always wondered how does a single fertilized cell can give rise to an entire organism with trillions of cells in a spatial-temporally controlled manner. So now with the single cell technologies, groups in UW and groups in broad BGA and groups all over the world can have generate many cell atlases that really provide the unprecedented view of the transition from the progenitor to mature cells across an entire organism. So what I'm really excited about is that can we actually go beyond just cell atlases to gain predictive models of the cell-fate transitions. So as we know, just because we know the theory of gravitation, we can use this theory to predict how the planet moves in the space over time. Similarly, I also wonder if we are actually able to derive the equations of cell-fate conversion. I believe we can also able to predict how the cells move in the gene expression space. So in my view, there are about two major efforts in the past few years that really moves towards this overarching goal of gaining the models of theories from the biology. The first one, in my opinion, is the pseudotemporal ordering. So with machine learning algorithms such as Monarch 2 and Monarch 3 that are developed in my PhD, we can leverage the gene expression similarity across the cells to learn a graph manifold, and then we can order the cells along this manifold so that we can quantify the relative position and branch of each cell in a dynamic process. So another very exciting development in the field is the introduction of RNA velocity. So in a very creative work, Lemona and others show that by leveraging the incidentally captured intron from single-cell RNA-seq, they can derive a measure of the time derivative of the spliced RNA or RNA velocity for each gene in each cell. So they can then project this high-dimensional RNA velocity vector to low-dimensional space to reveal the local dynamics of each individual cell. So RNA velocity is really one of the landmarks in obtaining the kinetic information from single cells. Nevertheless, both pseudotime and also RNA velocity methods are limited. Almost 4 years ago, when I was thinking about this problem, I wondered, can we actually go beyond the discrete RNA velocity vectors to derive a continuous equation of cell-fate conversion? And importantly, it's obvious to me that once we have such an equation, it will open doors to many fundamentally different analyses. For example, we can use it to predict the cell fate for each individual cell. We can also use it to resolve the regulatory network and predict the optimal reprogramming path and identify the key transformation factors. Lastly, they can also help us to perform in silico perturbation predictions. So in the following, with the intention to answer the question of building predictive models of a single cell spatial temporal kinetics. I'm going to first walk you through some data and experiments, and then I'm gonna also tell you how we can use how we can actually learn the equation with our Dynamo framework, followed by some examples that I hope will be illustrative of how this method can give us very fundamental insights. Finally, I'm going to close with my recent efforts in extending the predictive modeling to spatial transcriptomics and others with the spatial framework. So let's start with the data. So it turns out that to base the model the spatial temporal, that means the temporal kinetics, the ideal data is the metabolic labeling enabled single cell RNA-seq, such as the SCNT-seq. So we use the SCNT-seq to generate a data set of a human hematopoiesis. So we then also developed the tool Dynamics to process this data set to obtain new total spliced, unspliced RNA for each gene in each cell. With the data, then we can perform spliced RNA velocity with this data of the S3 velos dynamic model. Interestingly, the splicing RNA velocity reveals an overall pretty messy RNA velocity flow, and also problematic transitions from like the mature cell type such as megakaryocytes, erythroid, basophil, back to the progenitor. On the other hand, when we use the labeling data, Dynamo automatically reveals a smooth hierarchical transition from HSC into the GMP leg and also MEP leg cells, which then ramifies into all kinds of terminal cell types. Just really nicely recapitulate what we has known about the human hematopoiesis, indicating that the metabolic labeling based data can give us better results comparing to the splicing data. Another thing I want to mention is that because we have the labeling data, so we do know how long we label the cells. Therefore, the velocity from the labeling data is absolute instead of relative. Now with this data, I would like to tell you how we can develop, how we can learn the models of gene expression kinetics. Specifically, we use the dynamical systems approach as a foundation for understanding cell state transition. So under this perspective, each cell can be represented as a point in a high-dimensional state space where each dimension corresponding to a gene, such as X1, X2, X3, these three genes. The time derivative of the gene expression state, X dot, is a function of the underlying gene regulatory network, Fx. For example, in this cell S2, we have a complicated network involving thousands of genes. Although complex, such network may include characteristic motifs. For example, the mutual inhibition and self-activation network motif, such as the P1, GATA1 motif that play key roles in hematopoiesis. Assuming a noiseless system, the regulatory network function Fx will determine the trajectory of the cell fate xt. In this particular case, all the three cells state S1, S2, S3, they're coming from different initial cell state, but converge into the same terminal cell state, or attract A1. So since we really don't know the details of the complicated gene regulatory network, we would like to learn it directly from the single cell data. Fundamentally, this is a machine learning problem. So where we have some gene expression state X and some local dynamics of the gene expression state, or the RNA velocity, or dx over dt. Such data can be either measured with the conventional single cell RNA-seq or the 10 resolved metabolic labeling enabled single cell RNA-seq. The only difference here is that this later method can give us a real and sometimes more accurate RNA velocity. So with this X and the dx over dt, we can then use it to learn a continuous function. Once we learn the function, we can use it to predict the trajectory of each individual cell. And then use this function to perform novel analysis that other tools, including like velocity or SCVelo, cannot perform. In the following, I'm gonna highlight a few non-trivial predictions that, as I mentioned, other methods cannot achieve. The first one is about the least action path, or the LAP or lab predictions. So as we know, ever since the discovery of the cellular reprogramming, it has become a fundamental question in biology to predict the optimal path and transcription factor combinations so that we can convert one cell type to another. Previously, I used the LAP method to make such predictions, but only with the simulated system. Now, since we are able to actually learn the equations of cell-free conversion from single cell data, I wonder whether we can use it to also predict the optimal path on the real data. In the context of the hematopoietic system, we do know the key transcription factors for many of the developmental processes, as well as the reprogramming and the transdifferentiation events. So we would like to apply this method to reveal the to firstly validate the previous long transcription factors for each of the transition, and then we can use if we validate this method works on this previous known conversions, it's also possible that the prediction on the unknown transition will be accurate. So to understand how the lab method works, let's say if we want to start from the HSC to make the cell to become the megakaryocytes. So in theory, you can go from firstly into the erythroid, then to the megakaryocytes, but that's not really likely because some parts will be more likely than others. So the lab method will use a sophisticated approach to identify the optimal path that has the highest transition probability and smallest action. In this case, this first path is lab. So here I'm gonna quickly show you that we are able to accurately prioritize the known transcription factors for each of the reported transdifferentiation experiments. And you can see our priority score is pretty high. Most of them is above 0.8, indicating the high accuracy of our approach. Similarly, when we take all the known conversions and perform RAC curve analysis, we show that the AUC is around 0.85, again indicating the high accuracy of this approach. Given the success on validating on the existing reports, here I'm going to also show the RNA kinetics for the three top transcription factors along the LAP for the full transition matrix. As you can see, we also predicted the key transcription factors for just unreported conversions. And I believe it will be interesting in future direction to validate the genes and their kinetics from the juice predictions located in the yellow boxes. So here I'm going to also demonstrate the predictive power of Dynamo, highlighting one perturbation prediction of the SPA1 or PU1 GATA1 network motif. We know that PU1 GATA1, they mutually inhibit each other, will also self-activate. It behaves like a seesaw model. In particular, we found that if we suppress GATA1 expression, this will then break down the balance, and the SPA1 will then win, and cell will then move toward to become GMP-like lineages. And as you can see from our prediction with Dynamo, the cells indeed move from erosory MEP-like cells to become neutrophil and monocyte cells. This is nicely consistent with the previous biochemistry experiments. It's also worth noting that our in silico perturbation is highly efficient and combinatorial, so that you don't really need to do any experiments to make genetic perturbation predictions, and this will provide an important complement method to use a genome-wide perturb-seq experiments that reported from our lab. Finally, I'm going to close with how we can extend this predictive modeling to the spatial transcriptomics and others with our new spatial framework. We know that the cells don't live in a vacuum, but constantly receive signaling from neighboring cells. However, existing single-cell method will dissociate the cell or tissue or organ, rendering the complete loss of the spatial context of cells. Therefore, it is of great interest to profile the gene expression in situ and ideally even in 3D space. With such data, eventually we would like to build the SpatioTemporal models for most embryogenesis. So what do I really mean by in silico SpatioTemporal models? As an illustrative example, let's say if we wanted to understand how the 5 digits in mammals form during the organogenesis, and assume I'm able to actually profile the gene expression kinetics of the key genes like SOX9, BMP1 to over time and space. What I really want is able to learn the functions of this process, as I write down here. In many ways, as you can see, this is similar to the vector field learning that reported in our Dynamo paper. But for this digital formation, we really learn, what we really learn is the partial differential equations, because it also includes spatial diffusion, such as the BMP and the window signaling. So with this, towards this overarching goal of learning the SpatioTemporal model, we then develop a new tool called Spatio, which is just actually released today. If you're interested, you can go to download it and play with it. So Spatio support analysis for for many popular spatial transcriptomics, including stereo-seq, slide-seq, vision, nanostring, and many others. It also has functions to perform single-cell segmentation, cell type clustering, spatial domain analysis, and the ligand receptor interaction, and many more. Importantly, Spatio is equipped with sophisticated methods for building whole body 3D models of embryogenesis by leveraging the stereo-seq. Here, I'm gonna highlight how we can use Spatio to build the whole body 3D models of the drosophila embryo. So with stereo-seq, we can then perform continuous slicing of entire drosophila embryo, and then we can map the gene expression in situ. Then we can apply algorithms that learn the optimal affine transformation to enable multiple slices, which then can be able to use to reconstruct the original tissue 3D structure. So once we enable the chips, then we can use it to build the whole body 3D models. So firstly, cells from the same embryo, where the coordinates of each cell is named and also annotated with different cell types, shown as different color here. Then we can use the point cloud to create the surface. and then we can fill the surface with the volume, basically by inserting the work cells inside the surface. Later, we can also create volumes for each tissue and then embedded different tissues such as like CNS, muscle, and so on in this embryo. So it's also clear, we clear that even with this serious selection and also 3D reconstruction, to reconstruct the embryo will undoubtedly has gaps in the middle and also missing for a lot of cells. Therefore, we also build a deep learning framework to learn a continuous gene expression model. For example, here we are the deep learning will take the spatial coordinates to output the gene expression at any point in these 3D models. So as an example, you can see here we have the gene expression on the space with a deep learning approach. We are able to learn the continuous gene expression on the entire 3D model. Ultimately, we hope to use spatial to understand how the tissue expands, shrink, or migrate, diversify, or converge. We are currently wrapping up a paper on these topics, and we will release it pretty soon. So with that, I'm gonna thank Jonathan for all the support during the past few years, which made it possible for me to work on the very exciting project for Dynamo for my postdoc. I also want to thank Chen, Joseph Anchi, and many others from the Weissman lab for all the helps on performance experiments and the discussions. My great thanks to Yan Zhang, Jianhua, who contributed enormously to the theoretical part of the Dynamo paper, which and also Yan is the co-host for that paper. So of course, I also wanted to thank my PhD advisor Cole for his wonderful support during my PhD to develop Monaco 2, Monaco 3 that I briefly talked about. Obviously, I also want to thank many collaborators, like, including Vijay, Eric, Jorge, Shaya, Spiros, Hall, and especially I would like to thank for this Spatio project. I would like to greatly thank the close collaboration with the BGA research, especially Shipin, Longqi, Yinji, and many others. So I want to end my talk with this analogy of searching a unifying theory in astronomy. That's like astronomers who like to understand the planetary movement. Biologists also want to understand how the spatial-temporal kinetics works. With big data that generated with stereo-seq and other methods, we can also derive data-driven equations with Dynamo and Spateo. Eventually, this will lead to theories of biology. And in many ways, this is similar to collect the big data of planet movement, which then leaded to the discovery of empirical law by Kepler and then the theory of gravitation by Newton. Right, that's all. So a little bit over time, but I'm gonna stop here.
Thank you.